Let me now introduce the Turing-Roche knowledge series, whose aim is to bring together members of the Turing-Roche networks and the wider scientific community to hear different perspectives on topics in data science and also to build new connections. To get us started, let me go through a couple of slides to give you an intro to the partnership. So yeah, as the name suggests, it is a partnership between the Alan Turing Institute and Roche. I guess most of you are familiar with at least one of these organizations. In case you're not, the Turing Institute is the UK's National Institute for Data Science and AI, and it's built around a network of university, industry, charity, and government partners. Roche is a leading healthcare company with a long-term interest in personalized healthcare, and we're one of their growing number of academic partnerships. And the partnership was launched in 2021. It is a 5-year mission, and it's summarized by our partnership North Star. The goal is to collaborate to achieve a better understanding of patient and disease heterogeneity, essentially why patients respond differently to treatment and why these diseases can manifest in different ways in different people. So we're doing this in a stepwise manner, as illustrated on the right. So not on the drug discovery side, but we are focusing on developing new methodologies and working on knotty challenges that occur in large health data sets. So we have split our North Star, focused on research into these 3 different themes: structured missingness, to deal with missing data, in particular when it displays in a structure; predictive modeling, to construct models that are robust to uncertainty and heterogeneity and that they are transparent; and multimodal analysis to effectively combine different data modalities to improve clinical care. Our core partnership team is pictured here with representatives from Turing and Roche, which is expanded by the research associates funded by the partnership, enrichment students, and community scholars. And you can find all the people involved in the partnership on our webpage. And now I will leave the floor to Deepak Parasar, the deputy lead of the partnership, to introduce our speaker.
Thanks very much, Maria. Welcome everyone to the session, and it's a real pleasure to welcome Professor Robert Beckman from Georgetown University to the Turing as well as the Turing-Roche partnership. Professor Beckman is a multidisciplinary academic at Georgetown University in Washington, D.C. He's a clinical oncologist and at the same time a professor of biostatistics, bioinformatics, and biomathematics, which means that he can actually talk the same language as a clinician and also as a data scientist would. So that makes it very handy to have conversations and collaborations. Professor Beckman has had quite an illustrious career following education in medicine at Harvard. He's had training in Stanford, and I believe also postdoctoral work at University of Michigan. Of particular relevance, especially for the Turing-Roche partnership, is that he's also spent about 18 years working in pharmaceutical industry. And then making the move back to academia sort of speaks volumes of his sort of passion for furthering research in cancer, and most of his research has been in cancer evolutionary guided precision medicine. And he's also a sought-after speaker in the community on basket trials, which is sort of quite a novel sort of ways to design trials, especially in oncology and rare diseases. So it's a real pleasure to have Professor Beckman here, and we look forward to your talk on dynamic precision medicine.
Well, thank you, Deepak, for that very kind introduction, and thanks to the organizers for having me here. And as I said to someone in the hallway, we all look way up to Alan Turing, so it's quite an honor. I'm on fit. You permitted to speak here. So this is sort of this slide really Deepak has covered in his kind introduction, but I'm going to present stuff that is far in the future for oncology. There are real challenges in translating some of these ideas, and I don't want people to think I'm unaware of those challenges because I spent 18 years in industry doing clinical trials of experimental drugs in cancer, and so I understand that what I'm trying to do is challenging. On the math side, I'm self-taught, started at a young age. For 5 decades, I've been practicing math without a license, and I have some ability to apply what I know, but I don't have the depth of that you all have in the audience, having done it full time for so long. So, but anyway, so I'm going to speak about genetic instability in cancer and implications for drug resistance. And it turns out that although you're familiar with that problem is perhaps worse than people have realized. And I'm going to I'm going to talk about dynamic precision medicine, or DPM, which is a new approach to the precision therapy of cancer, which I hope would be the next small step beyond the current precision medicine approach, which, despite being a relatively crude model, has provided significant patient benefit. And I'm going to talk about simulation studies, lab studies, challenges on the path to clinical transformation, clinical study designs, and possible collaboration through these partnerships with Turing. So people in the audience probably know cancers have multiple genetically distinct subclones, and they undergo continuous evolution. I do want to make the point that it's almost inevitable that some of these subclones will have mutation in DNA replication or repair genes required for chromosomal instability, checkpoints, and these, if they have mutations there, they're actually going to evolve more quickly. They're going to not maintain their genomes as pristinely as wild-type cells. And these subclones are termed hypermutator subclones. And whereas some of them may mutate themselves to death, some will emerge rapidly with simultaneous resistance to multiple non-cross resistant drugs. And in simulations, these hypermutator subclones, which are not emphasized as much in other models of cancer evolution, these are therapeutic priorities. They're very dangerous. So I'm going to describe my 50-year collaboration with Larry Hill, who's shown on the left hand and center picture. He just recently retired. He's in his 80s. So I started when I was a freshman in college working with him, and was his theoretician for a very long time, almost 50 years. And his influence on me is surpassed only by my father. This is his grandson, Brendan Korn, who did the bioinformatics for this work. So Larry was probably the first person to say that tumors are genetically unstable. He said that in the 1970s. And he's a very eminent person, privileged to work with him. From his lab, we actually had his people invented duplex sequencing, which is a different approach to DNA sequencing, which sequences both the Watson and the Crick strands simultaneously, and will not call a mutation unless there are complementary changes in both. And this is too expensive to do for the whole genome, unfortunately. But it is extremely accurate. As one technical error in greater than 10 to the 7th bases that are evaluated. And as a result, you can look for very rare subclones. You can look very deeply. So on the left, you see sequencing a depth of 10,000, it's P53 gene, and you're basically in the noise. And this is the sequencing that is used in clinical data collection. And on the right, you see the same depth of duplex sequencing, and there is no noise, and there is one peak, which is the positive control of the dose mutation. So we looked at colorectal cancer patients at diagnosis from colonoscopy, and did deep sequencing and I did some mathematics on it. And what I would say, if you look at the iceberg on the left, so the field is mostly involved in working on the iceberg part that's above the water, which is very important. These are the clonal mutations that are present in 10% or more of the cells that have been selected because they're cancer drivers, and they certainly inform drug selection and so on. But below the surface of the water, there's a hidden cancer genome with an enormous reservoir of diversity that didn't matter when the cancer was evolving in the absence of drugs. But it's a secret weapon when you start to apply drugs. And so if you look at the figure on the right, what I suggested to them is that they sequence at increasing depth, which is on the x-axis, and look at the number of sites in a small sequence of DNA. We had 13 kilobases of DNA only, that look for how many uniquely mutated sites there are. So if something is mutated 30 times, that still only counts as one. Another one is kind of mutated only once, that counts as one. How many sites can you see as you get more and more sensitivity by sequencing deeper and deeper? So you could hike into the Grand Canyon, and as you go deeper, you're going to go further into the past. But as you go deeper in DNA sequencing, you go closer to the present because you're further out on the evolutionary branching tree. And so from theory, we could show that if these mutations that are below the water at less than 10% variant allele fraction, if these mutations accumulate according to this curved line, if they're positively selected, and according to a straight line, if they're evolving neutrally without selection for or against them. And this is the data. They're evolving neutrally. This is from multiple different cancer patients who had slightly different mutation rates. And if I showed you the individual plots by patient, they fit perfectly. It looks like it's fake, but it's not. So we published this in PNAS in Clinical Pharmacology and Therapeutics. And recently the theory again was reviewed in NPJ Systems Biology and Applications. And the messages are that subclonal diversity is much greater than previously believed. And this could not be detected by conventional sequencing. The mutation rate is about 100-fold greater than previously believed. And based on the mutation rate and the number of cells in the smallest lesion that you could see on a computed tomography scan, you can easily show that resistance, well, any mutation that you want, unless that mutation kills the cell. And there are very few single mutations that kill the cell. But if any mutation that you want is going to be present in multiple cells, and at least one. And if there's any mutation anywhere in the genome that gives you resistance to the miracle drug that you just invented, it's already present in everybody's tumor. But we give combinations. Combinations have toxicity, and not all drugs are non-cross resistant. But anyway, here's another implication which is not published yet. This is something I hope to write up in the next month or two. But we have shown from the theory that the vast majority of clinically relevant resistant subclones are present at quite a low level and would be undetectable by the current sequencing methods. So this raises an interesting philosophical point, which is a whole other discussion, which I won't go into. But when we do laboratory experiments, we have engineered cells with mutations. Then somebody says, well, how do you know this mutation is relevant in humans? Let's go look in the databases. Well, it's fine to look in the databases, but you know what? Those people were sequenced with a technique that can only pick up 0.01% of what's there. So if it's not there, especially since we've already shown that everything is there, I don't get worried about it. Grant reviewers get worried about that. That's a different challenge. So we had we developed a theory which I'm going to discuss briefly. On the x-axis is the size of the tumor as it's increasing over time on a log scale from 1 cell to 10 to the 10th and beyond. And on the y-axis is the variant allele frequency of the average mutation that is formed at the moment when the tumor is that size. So as the tumor is larger, the variant allele frequency is going to be less because you're further out on the branching tree. And this research was done simultaneously with some brilliant colleagues in the UK, and they have the yellow line, that's their theory, and ours is the red line. And I'm going to explain the differences. Ours, we had to wait 3 years to publish ours because we had a potential scientific integrity issue that had a result. But the work was actually done at the same time, and I was unaware of their work. And there are quite a bit of similarities, but the important difference is here. So with their work, this is a log-log plot. As the tumor gets larger, the subclones keep getting rarer and rarer in inverse proportion. But what we found is that as the tumor gets larger and larger, there's an asymptotic limit of the variant allele frequency that is determined by the, as you might expect, by the physical chemical ability of the DNA replication machinery that just can't keep getting at a limit to its accuracy. And so our math describes the asymptotic approach to this limit. And the reason why this is important is, you only diagnose the tumor when it's about this size. And then it's about 3 logs greater by the time the patient dies or more. And there's a big difference in the amount of diversity that you see here in the area under the curve. So even quite apart from the fact that the mutation rate is 100-fold greater than we thought, it gets worse in a diagnosable tumor, and it gets worse as the tumor gets larger. Things get much worse as the tumor gets larger, and we need to minimize diversity as early as possible before it gets out of control. And with all due respect, it doesn't matter how many targeted agents Roche comes up with. At some point, when a patient is terminal, it's too late. So dynamic precision medicine is not the complete answer to that, but it's trying to be the next step in a progression of better answers. And dynamic precision medicine explicitly considers, and DPM explicitly considers subclonal heterogeneity, tumor evolutionary dynamics. It makes decisions ahead of time based on probabilities and risk states before you can actually observe something. Typically, by the time it's observable, it's going to be too late. And it adapts therapy very frequently, as often as 6 weeks. These are called moves, an analogy to chess. Like a good chess player, it tries to think ahead and do long-range planning. And the goal is not just short-term tumor shrinkage, but prevention of multiple resistant cells and to prevent moderate to long-term relapse and enhance survival. And you may actually have to give up something in short-term tumor reduction to achieve that. So current precision medicine focuses on average molecular characteristics of a bulk specimen. Dynamic precision medicine considers minority subpopulations. Current precision medicine focuses on the current biopsy. Dynamic precision medicine is trying to work backwards from the end game where the patient becomes terminal. And we have this thing called the penultimate treatable state, which is the last moment before the horse escapes from the barn. Then you want to close the barn door before the horse escapes. Try to think several steps ahead. Instead of treating with the same therapy until the patient worsens, we adapt frequently and proactively. And we try to perform mathematical optimization across the entire treatment course. And, dare I say that AI and ML might have quite a bit of relevance to our ability to do that. We have our approaches right now, but they could be improved. This is the toy model that we've been working with, which looks very simple, but when it's applied to this problem, it rapidly becomes complex because of the combinatorics. So we have 4 cell types, which are really mixtures, but we treat them as sort of average populations. The S cells are, I'm sorry, this is a 2-drug system we've looked at 3 drug systems. The expert clinicians would pick the drugs. The drugs should be, insofar as possible, non-cross resistant. And, I don't want, 2 chemotherapy drugs that are both substrates for the MDR efflux pump that you can become resistant in one step to both of them. So and actually a drug may itself be a combination. So a BRAF inhibitor in combination with a MEK inhibitor would, that combination is trying to kill a certain type of subclone. That would be considered drug 1, even though they're 2 molecular entities. So you have these 2 non-cross resistant drugs or combinations. S cells are sensitive to them both. R1 cells are resistant to drug 1, R2 cells are resistant to drug 2 only, and R1-2 cells are incurable because they're resistant to both, and the goal is to prevent their formation. And then they all are growing exponentially, and the arrows represent, sorry, I'm pointing, not only I can see it, the arrows represent genetic and stable epigenetic transitions between these phenotypes. Cell growth and death are affected by drugs in a dose-dependent manner. That means when you give combinations, if you may have to reduce the dose because of toxicity, you have a trade-off. On the one hand, you're giving more diversity to your therapy, but you don't have the optimal dose, perhaps. And because we adapt very frequently, you don't have to give them all at once if you can. So maybe you're going to find someday that you need to give 20 drugs and you can't give them all full dose. But if you adapt frequently, you can get dose intensity of all of them in. It can be partial resistance, and importantly, patients have a mixture of cells which evolves over time. So I'm going to go over this very quickly and not present all the details, but this is a case that I contrived after writing out the differential equations of the system. And this case basically shows, for current precision medicine, the drug therapy is across the top. The patient presents with 10 to the 9th cells, the total cells are the blue line, and you give the best drug for these cells, and the tumor shrinks like crazy, and you can't even see it anymore, and everybody's happy. But below the level of detection, even by PCR, there was a teeny-weeny subclone here that's already resistant in the red line. And it grows back, and the patient relapses at 15 months. Then they repeat the process according to current precision medicine. And it turns out that drug 2 is the perfect drug for that. And they give it, and again, everybody is celebrating. But what's happening here below the level of detection is that these singly resistant cells acquire double resistance. And this double resistant clone is growing out. And at 28 months, there's nothing more to offer this patient. And this is what we see. And I've treated patients, and the benefit gets shorter and shorter with each subsequent line of therapy. But if you understood that these red resistant cells might be present, and for the particular input parameters of this virtual patient had a very brief pulse of the drug that would kill off this minor subclone. And meanwhile, the tumor size is sort of slightly increasing, but it's not enough to be an emergency. And then the doctor switches to a combination and the patient is cured. And this obviously is going to be different for every virtual patient. And the expertise of the doctor will tell you when you can afford to just watch the tumor with stable or slowly growing disease for a brief period. So here's a graphical representation of what I'm trying to explain here. In the dotted circle is these are all the phenotypic states which are treatable by the available drugs. And within that space, there's a big yellow circle, which we call the initial predominant state, which is the main subclone that is present in a bulk sample that has a BRAF mutation and has some other molecular characteristic. And then there's this smaller subclone that is perhaps even undetectable or a minority, which we call the penultimate treatable state, because it is closer to the to escape from this envelope of treatable states. And it may even be a hypermutative subclone, so it's evolving more quickly with a heavy black arrow. And once it escapes, it forms an end state, which probably is self-explanatory, unfortunately. And so the question is, so you can't really ignore this big yellow circle because it's causing symptoms, it has other, and plus it's generating new variants. And so you would like to cytoreduce the tumor. But this little red guy is also a priority. So DPM is a method for figuring out every 6 weeks whether you should give the therapy that's optimal for the yellow circle or the therapy that is optimal for the red circle, if they differ. And every 6 weeks you reassess this. So DPM is designed for use with multiple non-problematic agents. It focuses on preventing multiple resistance and late relapses, and different subclones may evolve at different rates, which is, among other evolutionary theories of cancer, this is somewhat unique. So I designed and contrived the example that I showed you when I was privileged to be a part-time member of the systems biology group at the Institute for Advanced Study in Princeton. And there I met people who were not part-time mathematicians like myself, who were extremely good at mathematics and computer science. And one of them was Chen-Sheng Yang, who's shown in this picture. He's now a professor at Academia Sinica in Taiwan. And we're in front of the art museum there. And Chen-Sheng set it up so that we could do high performance computing simulation of 3 million virtual patients that had various different characteristics to prove that to find out if my contrived example was a fluke or not, and it wasn't. So we looked at strategies, where a strategy, I think for a computer science group, this wouldn't be a surprise, but clinicians think of a strategy as a particular sequence of drugs, whereas we're thinking of a strategy as an algorithm for individually assigning a patient to their optimal sequence. This is probably not surprising to computer scientists, but just clinicians think a different language. They speak a different language. They think a different language too. So, so we looked at current precision medicine strategies, which are treating with the best drug for the big yellow circle until the patient gets worse, and then reevaluating. And we looked at 5 dynamic precision medicine strategies, which used the differential equations to predict the best strategy for the yellow circle and the best strategy to prevent the black circle from occurring. And if they were different, we had heuristics to prioritize between the two, which were different among the 5 flavors of DPM. We did this every 6 weeks. We started out with 30 million virtual patients, but a lot of them were trivially easy, or they only had one drug that worked, so there was no strategic dilemma. And we ended up with 3 million, but they had different initial populations, different growth rates, different transition rates, different levels of sensitivity resistance to drugs. And these were, at the time, told from my personal knowledge of growth rates of cancers that I had seen over my career, and also from my work with Larry Lowe, we did a lot of DNA enzymology to talk about mutation rates at the enzymology level. But of course, in some future iteration, we would like to do this by looking at national databases and not just using what's in my head, which is going to be limited, but we ended up with 30 million parameter combinations. We explored the 3 million non-trivial combinations, and this was meant to be a sensitivity analysis across oncology. And this is the Kaplan-Meier survival curve. The blue line is current precision medicine. This actually looks a lot like what we're seeing in oncology, and which is to say, very few patients live beyond 5 years, and about half of them are dead before a year. And the other lines are all the different flavors of dynamic precision medicine. And they in silico, they double mean and median survival, and they result in a greater than 20% long-term survival compared to virtually none with blue lines. But of course, this is in silico, and we had this result a long time ago, and clinical translation is a problem here. And part of the clinical translation could certainly be helped by data science and AI expertise. Some of it has to happen in the wet lab. We do have clinicians and laboratory people who are working with us who have become excited about this in breast cancer, melanoma, colon cancer, and leukemia. But there's still challenges for sure. So we learned that rare cells matter, that hypermutator subclones are therapeutic priorities. We've also learned that giving all the drugs at once in simultaneous combination is not always the best answer. Sometimes it is, but if you have to reduce the dose when you give multiple drugs at once, then you have a trade-off. And so here we have, I'm not going to go into details, but this is a 3-drug simulation, the yellow, green, and blue drugs across the top. And you can see that this algorithm gives a very complex pattern where there are combinations and pulses of monotherapy. And this combination keeps the patient alive for several years before the tumor escapes. And if we take the same virtual patient, we have another very complex combination, again, another very complex pattern with both combinations and single agent pulses. And this more powerful algorithm cures this virtual patient. But this powerful algorithm does need more data. So I'm not sure I'm ready to, go to that for it, honestly, because that not having data from the patient is going to be a problem. Anyway, we have laboratory studies ongoing. This is our collaboration in breast cancer with Rebecca Riggins on the right as the PI, and Shaima Banasi was her postdoc, now has her own lab. And I'm just going to show you one thing that they did. They showed that when you select for resistance, you select for hypermutators. So that in fact, as we go through a patient's course and we select for subclones that are resistant to the drugs we're giving, we are also accelerating the rate of evolution. That's because these hypermutator subclones are more likely to have pre-existing evolution resistance and more likely to acquire resistance. And so here there's fluorophores that have been put into the cells. These are breast cancer cells that were sensitive to estrogen deprivation that were evolved to become resistant to estrogen deprivation. Same thing on the right, parental and resistant. And the red fluorophore is in every cell, but the green fluorophore is only in cells that mutated a defect in the green protein. And so there are more green dots in the resistant cells. And if you do the DNA sequencing, you find out that there are more insertions and deletions in the resistant cells. And in one of the situations, there are also more single nucleotide variants. Some of you may recognize the gentleman in the middle there who I could also give a wonderful introduction for, but I know I'm running behind on time. And on the left is Dr. Matt McCoy. He's an assistant professor at Georgetown who does a lot of the computer science for us right now. And on the right is Wei He. He's a postdoc. And so what's the problem with DPM, and why hasn't it revolutionized oncology yet? Because it's really hard to translate. And it requires information at a deep subclonal level. And to do it ideally would be extremely expensive, and no patient would consent to being biopsied every 6 weeks. And unless they have leukemia, looking in the blood is going to be have sensitivity issues. But meanwhile, Deepak and I were thinking about clinical trial designs, and we couldn't come up with a clinical trial design that would prove that we had the optimal 40-move sequence anyway. So we said, how about the first 2 moves? If you just did 2 moves of DPM, would that help at all? And I thought it wouldn't. And I'm proud to say that I was wrong, because it was really great. On this survival curve, the red line is conventional precision medicine. The green line is if you do dynamic precision medicine for 5 years, 46-week moves. And the blue line is if you just do DPM for 2 6-week moves, 3 months, and then you go to less invasive, cheaper, conventional precision medicine. And you can see that the blue line is nearly as good as the green line. There's not much daylight. So it's like child development, the early times matter, you know. And furthermore, if you look at the virtual patients who are most similar in outcomes between the blue line and the green line, and you say, I want to select them as my clinical trial population for pilot study, those patients have a benefit level which is just ridiculous. Even I don't believe this, but you know, let's say it's, you know, even if this is, you know, 80% wrong, it's still spectacular. So this would, we call this the evolutionary classifier to help us select patients who would benefit. And this paper recently got accepted in Journal of Clinical Oncology, Precision Oncology is in press. And this allows us to go into the pre-surgical setting in solid tumors, so-called neoadjuvant setting, where somebody has a breast tumor and they get some kind of therapies for 3 months before the surgery is done. And we could get a biopsy at diagnosis, we get a biopsy at surgery, and we ask the patient for one optional biopsy in the middle. And I can tell you from my experience running clinical trials that if you have a good rationale, patient will consent to one additional biopsy. Not 40, but one. And so the cost and tissue requirements and patient inconvenience are dramatically reduced. And we could use this to govern therapy in the neoadjuvant setting, or we could say we want to do fancier experiments with longer turnaround time. Then we use this to govern the first 3 months of the post-surgical or adjuvant period. And so I've already mentioned this, and I know I'm taking a long talk, so I'm going to just say that the data requirements for DPM are quite challenging. And in particular, we are interested in measuring rates, not just static states. And the current community doesn't even measure those at all. So we would need a massive change in the community to even collect this kind of data. And we need more sensitivity, and everything's listed there. Ideal is the first bullet, real is the stuff below. And so what does this mean? So we are going to be dealing, since Maria said the Roche partnership is interested in maximizing situations where you don't have ideal data. Had a situation where raising my hand. And we need help with this. So we have done what we call misspecification simulations, where we create a situation where there's a hypothetical doctor who's given incomplete or inaccurate or noisy information, and she or he makes the best decision using the principles of DPM based on their inaccurate data, and the system evolves according to the ground truth. So we have looked at the impact of the wrong sensitivity to drug 2, which is usually a late line drug that DPM will be recommending earlier, and there's no information about its clinical performance in earlier lines. And we asked, what if we have underestimated that? Will DPM falsely look good or bad, depending on just how good this drug is? And the answer is to some extent, yes, but it's remarkably robust. And also the clinical trial design that we're simulating for this, which Deepak and I have worked on, has pretty good statistical properties. And Deepak and I hope to finish writing that up this week. And we'd like to submit it to Nature Cancer if it turns out to be appropriate for Nature Cancer. Then there's the problem with the limited sensitivity of detection of rare subclones. And I pointed out that the sequencing is not going to detect that. And this is, I'm just going to show the slide here. On the x-axis is the smallest subclone you can detect. On the left is better. That's where you could detect almost any subclone. And as you move all the way to the right, that's the insensitivity of the real methods that we use on patients today that are economically feasible. And then on the y-axis is the hazard ratio Smaller numbers indicate more benefit, less than one, you have less risk of death. And again, the bottom is the best and the top is the worst. And if you assume that when you can't measure a subclone, it's absent, then you get the red line and your hazard ratio goes from 0.5 to 0.75, which is still pretty good, but it might not be worth all this trouble. 0.5 is worth all this trouble if it's real. But that's if you assume zero when you can't measure something. But if you assume a probability distribution function rather than zero, a probability distribution function between zero and the limit of detection, which we have derived based on evolutionary theory, you get the blue line, and the blue line preserves most of the benefit. And you can use other generic statistical distributions. They help a little bit, but not as good as the one that's based on evolutionary theory. I want to just briefly mention that we have a searchable database of millions of trajectories of virtual patients that we can search by various criteria and do various analyses that we would like to make part of someday of a digital twin interactive interface. And this is certainly an area where additional where a partnership with Turing would be ideal for us. And so in terms of collaboration opportunities with the Turing, I think there are two main areas. One is we have this incomplete information on individual patients. And what we want to be able to do is take all the data like the UK national database and other things, and we want to be able to intelligently search this data and find out what is the probability distribution for the population of which the patient is a member. That is, let's say I'm going to speak like a Bayesian, that is a prior distribution. And then we have some additional information from the patient that gives us a posterior distribution. And the information from the patient might not be enough about that prior. Also, we will need to really create a digital twin sort of model and user-friendly interfaces for healthcare providers who may lack quantitative skills. There's that guy again. So we're working on clinical trial designs. And I mentioned that we could work in the neoadjuvant space with just a 2 months of DPM. And here we're talking about neoadjuvant breast cancer. We pick the patients who will benefit. We sort them into optimal sequences. And then after surgery, they just continue with current precision medicine. And then we look at a high risk group. How long is their relapse-free survival? Without a control group, this would be a small pilot study. Then we go on to the study design that we're hoping that Nature Cancer will find suitable. That is a randomized study where we stratify by the classifier the patients who we think will and will not benefit. And within each stratum, we randomize between DPM and conventional precision medicine. And then we have the more difficult problem of confirmatory trial designs that would prove that dynamic precision medicine works. And the problem is that there is still a chance that if we pick the use case, if we're lucky when we pick the use case, that these late line drugs we're going to be using earlier are so good that it looks like DPM is wonderful. And in fact, it has nothing to do with DPM. And similarly, we could be unlucky and DPM could be pretty good, but we picked the wrong use case and it looks like DPM is bad. So to have an unconfounded estimate of the value of DPM rather than specific drugs is a non-trivial problem. I've taken quite a lot of time, so I won't go into details, but I will say that Deepak and I are very happy to have the additional expertise of Martin Plummer from Warwick, who is sitting next to Deepak, and we hope to simulate the properties of the design we have and consider additional designs. And we could also consider designs that might benefit from AI, ML methods together with experts at the Turing or elsewhere. So in summary, DPM differs from current precision medicine and other evolutionary guided paradigms and may lead to striking improvements in outcome across a broad range of situations. It's designed for multiple therapies, aims to prevent moderate to late-term relapses, benefits from long-range planning, but yet can give benefit after only 2 months of therapy. We have a classifier for selecting the patients who will benefit. It's unique in identifying hypermutator subclones as the therapeutic priority. And we have novel clinical trial designs to test DPM in collaboration with Deepak and now with Dr. Plummer. I will close before I get to the acknowledgements with the hockey great Wayne Gretzky, who has summarized DPM far better than I could with this quote there. So here's some references. The red ones are perhaps more important. Here's some of the people. Here's more people. We need to acknowledge the Institute for Advanced Study at Princeton, the British Royal Society International Exchange Award, Oak Ridge National Laboratory, and also the Department of Defense Breast Cancer Research Program Breakthrough Award. And I just want to say in closing, there's some math there if you want to see it, but in any case, I just want to say in closing that for me, the earliest thing was finding my father's math books and falling in love with math. But then I went to medical school and I took care of cancer patients. And I took care of children with cancer. And I had to explain to parents that their child had cancer. And that was a very devastating moment for them. But it was nothing compared to when the child relapsed. Because by that time, they knew that all their friends who they had seen on the ward, when the kid relapses, there's very little hope. And they know that. And so what I would like to prevent is relapse and cancer in general. But I'm not going to whitewash it. Translating this is not easy. So thank you so much for your time.
Thank you so much. Wow, what an interesting presentation. Shall we get questions from the room initially? Please. Yeah, we can hear you. Yeah.
Okay, sorry, I came in late, but I was online, so I heard everything. So the question is, when you're developing these clinical trials, isn't it possible maybe to use surrogate diagnostic tools, companion diagnostics like cell-free DNA or circulating cancer DNA to be able to constantly monitor the cell's mutations?
Yes.
So it's actually more doable instead of going in to do actual biopsies.
Yes, so we actually want to do both. As you know, with circulating cancer DNA, quite a bit of the DNA is from normal cells. And so you have the sensitivity problem that I described gets worse. The other thing is that we're particularly interested in if a single cell has 2 resistance mutations, the drug 1 and drug 2 in the same cell, whereas in the cell-free DNA situation, we can't tell if the cells or not. But we are interested in that. And in fact, one of the people who is interested in collaborating with us, Catherine Panabier, University of Montpellier in France, she coined the term liquid biopsy. And she has some wonderful also circulating tumor cells, cell lines made from circulating tumor cells at different time points in the patient's time course. And we want to learn from those. So yes, we're very interested in that. All of the current technologies can certainly help, but we are still going to have very incomplete information. You had your hand up, I think.
Yeah, it's a very naive question. How many, I don't know what you call them, these subpopulations, these
Subclones.
subclones, how many are there?
Well, so it depends how you define them. So as you know, there are 3 billion bases in the genome. So do I have the wild type base at each of those 3 billion positions? Yes or no? The answer is 2 to the 3 billion. I guess it's sort of. But then which ones are going to be relevant, we don't know. And what I would say is people who want to give simultaneous combinations, I think what we already know is that from this analysis that we've done is that 2, 3, 4 drugs like people are trying today are not going to match the level of diversity that the cancer is offering. So unless people can come up with, I don't know how many drugs, but maybe it's 1,000 non-cross resistant drugs that they can give all at once, full dose in an enormous IV, and it's all covered on insurance, then we have an optimization problem that we don't have. We can't do what's really optimal. And so, so I think our goal cannot be a general solution for cancer. But what I would say is back in the 1950s, when people were just trying to kill the cancer faster than the patient, and then they were trying to use combinations, they benefited patients. It was better than nothing. And now we have what I consider to be a very crude static model of current precision medicine. That's the current state of the art. And despite its naivete, it has produced tremendous benefits for some patients. So here we're sort of making the next level of approximation. And we're saying, okay, here's where we are today. We know there's other stuff that we can see and stuff that we can't see. And we know the rate at which things are changing. We know the first derivative. We don't know the second derivative. We don't know, we're pretending it's all linear and independent, all this stuff, which is not true. and so ours is crude, but it is the next level of approximation. And our hope is that we can provide significant additional benefit with that next level of approximation. But in terms of the diversity, it's scary. Yes.
So in terms of the modeling, what kind of competition is there in the model between the subclones? Because I'm quite familiar with the work of, I don't know if you know, like Gate and B and the
Gate and B very well.
adaptive therapy stuff. So
we disagree on everything, but he's a wonderful man.
Yeah. So is there's some kind of competition between the subclones in this model as well?
No. So if you look at the literature, I can find you examples where subclones produce goods that cooperate with each other in cooperative communities, and also situations where they're independent. So what we've done here is say the default model is independence, because that allows us to look at, let's say, in the lower right-hand figure that I showed you with the three colors, yellow, green, and blue drugs, we pruned the tree and looked at a subset of 7 to the 40th 40 move sequences. And we didn't want to do competition there, but the fact is sometimes they're going to be competing, sometimes they're going to be cooperating, sometimes they're going to grow independently. And I believe in any one patient, all of those things are happening in different ecological niches, micro niches, in the thousands of visible and invisible mutations that eventually kill a patient. So if the math that we use is flexible, so if somebody would say to us, Mr. Jones has competition between subclones A and B, and 84% of his metastases, then we could put that in. But we believe it is not a good modeling is always a trade-off between the complexity and the clinical applicability. And we don't think that enough is known about that to just pick one game theory model and say that's the model. We're going to optimize in this model. Obviously, Gate B has done fantastic work, but we think that this is also worth the shot.
We have 2 questions from our online participants.
Okay.
Griffin Farrow is asking, do you see DPM being more effective for certain cancers than others? For example, prostate cancer is notoriously heterogeneous. Would it be less amenable to this?
Okay, so that's it. So one of the things that we're looking into, so this evolutionary classifier tells us who will benefit, but it's kind of a black box. We're trying to understand what are the properties of a virtual patient that would allow them to be considered a patient who would benefit based on our classifier. And one clue we have, which we're pursuing, is that DPM doesn't help in a situation where it's too easy, and conventional precision medicine can do just a great job. And DPM doesn't work where it's too hard, where it's too late to try to do this. And there's kind of a intermediate zone in which DPMs benefit. That's the hypothesis. So when we develop some math around that, and we're going to test it soon. And so if that's true, we could answer your question for a specific prostate cancer patient. What I like about prostate cancer is that it usually evolves over a long period of time. So an approach that thinks about the future might be helpful. You also have the PSA, which is not perfect, but you could use that as another endpoint. And Dr. Gatenby actually has some very interesting results in prostate cancer. But we're not working in prostate cancer because we have not yet found an excited collaborator. We have found excited collaborators in breast cancer, colon, leukemia, pre-leukemia, head and neck, and melanoma. And that's more than we can handle, actually. And what I would say also is that leukemia, we can get the more sensitive biopsy from the blood. That's what it goes in the blood. So we can get more information from leukemia. And we have a collaborator there who already is exploiting that to a great degree and just needs a mathematical approach to prioritize among all her results. And we are trying to find the right words to convince a funding agency to. And the problem with us is this is very speculative, very hard, right? So there's one conservative person on every study section, so we have to convince that most conservative person that they want to take the risk. So anyway.
On the cost effectiveness point, Aisha Mula says, very fascinating work. I'm curious about how these inputs have been integrated into cost effectiveness models and what has been shown.
We haven't done cost effectiveness models, and I think the problem that we have is that we're still exploring different technologies. We haven't done clinical studies. We have laboratory studies that are very early days. And so we don't know what technologies would be used. So it's hard to say. And also the effectiveness in the computer is fantastic, but it's only in the computer. So I don't think we're in a position to meaningfully consider that. But I think that depends on the healthcare system and politics and stuff. But if you go from virtually zero long-term survival to 25% long-term survival, that's what immunotherapy has achieved. And if we could have a similar impact, people are paying a lot of money for that. Anyway, I'm sorry, other questions?
No, these were the online questions.
You have another question? Yeah.
If I may.
Yes, of course.
So this is all pre-metastases stage 2 level. Anything stage 3, 4 is probably difficult to use because they're already metastasized and you don't have biopsies of those sites.
So the mathematical model is just looking at the total number of cancer cells across the body. And in fact, that is another reason. I didn't go into all the reasons why we don't fund a competition between the subclones, but competition between the subclones is only in effect once the lesions reach a certain size where nutrients can't easily diffuse in and out. And those large lesions create a lot of radiology data, but they don't kill patients. What kills patients is infiltration with a bazillion tiny metastases, all of which can, they haven't even called for blood supply in because they don't do, you know, so they're not necessarily competing in a resource constrained environment. But so we actually think of this as the total cancer in the whole body. But the neoadjuvant setting would allow us to get some biopsies in solid tumors and fits into this two-move paradigm that I mentioned. And so that's why we're thinking of it. But then you notice the clinical trial was in a high-risk subgroup that we assume they have microscopic metastases that have not been seen. And it's those microscopic metastases that we really care about in that scenario. I don't think we will be as good at shrinking the tumor before surgery because we're not going after the big yellow circle.
Well, you're not controlling for the systems biology of the immune system, the impaired metabolic system, the complete systems biology. You're not controlling for that in this model, right?
Not yet. Those are things we would like to add. We have just submitted a paper to NPJ Systems Biology and Applications where we talk about we combine this model with the model of transient gene expression and cellular plasticity without genetic changes. And that is still a tumor-centric model. It doesn't. We understand that we need to do all these things, but we're just doing them one at a time. Yeah, and we'd love some help to do them.
Okay, so you can see it on the box. Does the strategies use a specific type of therapeutic, small molecule, large molecule, cell gene therapy? And is the structure of the tumor considered? For example, 3D organoids are used in preclinical trials to better model the tumors and its interactions with other cells. Very interesting presentation, and thank you for coming out to share your research.
Okay, so first of all, as was pointed out by the other questioner, we have not yet addressed the complexities of immunotherapy and its very unique interdependence with the TME. And by the same token, we would, although we're interested in subclones that are sensitive and resistant to therapies, we may have to use as a surrogate. Do they have a resistance mutation or something else like that, which is more something than a targeted therapy? I think we could more easily work with targeted therapies than with chemotherapies. So I think that the easiest thing for us is targeted therapies. We have some of our collaborators, we have put out proposals to add immunotherapy to that would involve a lot of additional development work for us. And we would like to use combination of small and large molecules because we're really assuming that we can find 2 or 3 or more relevant non-cross resistant drugs. And a lot of the small molecules are all going to be kicked out of the cell by small molecule efflux pumps. So in breast cancer, we're working with a HER2 positive, estrogen receptor positive, double positive breast cancer. And we want to use trastuzumab and pertuzumab that is going to be outside the cell and not subject to efflux pumps. That's working on one signaling pathway. And then you want to use small molecules against the estrogen receptor pathway. And by using not only different pathways, but also different modalities in different cellular locations, we hope we can get some degree of non-cross resistance. This is work that is occurring in the lab right now. And Dr. Riggins and Dr. Benassi are PhD scientists. And when we get some preliminary results, we will involve clinicians. We have other cases where clinicians are just wanting to do a pilot study, and we're trying to figure out how that could be supported optimally. We certainly would not suggest more than a pilot study at this time because of all the work that still remains to be done.
Thank you so much, and thank you to our online participants for staying late.